Okay, so TB. So now we've gone from the most complicated thing we're going to talk about to slightly less complicated, but still way more complicated than any virus. So we're in a bacteria now, and we're in an acid-fast rod-shaped bacillus in terms of the type of bacteria that it is. Acid-fast is sort of its equivalent to gram-negative or gram-positive. It's acid-fast in terms of how it stains. It has this unique wax-rich cell wall composed of long-chain fatty acids and glycolipids. That's what makes it acid-fast. It's really just this little ball of fat, and that makes it actually really impermeable to a lot of drugs and a lot of things immunologically we would like to attack it with. It has 250 genes of this gigantic genome dedicated to fatty acid metabolism. So I told you coronavirus was huge because it was 30,000 base pairs, and here's TB at 4 million base pairs. 250 genes alone just targeted towards fatty acid metabolism. So just mind-bogglingly more complex than anything we're dealing with in the viral side. And it has a very slow 20-hour replication time. Anybody that's worked with TB knows that it's slow and difficult to work with. Wide array of a wide burden of infection worldwide, particularly in sub-Saharan Africa, South America, Asia, and many islands in the Caribbean and the Atlantic. 2.2 million deaths per year, and incredibly high burden of diseases and disability adjusted life years, so 45 million dailies a year. A third of the world, give or take, infected with MTB. Of those 2 billion people, 10% untreated would have a risk of developing disease following infection, with mortality up to 50%. There's 8 million new cases per year, 1 new case every 4 seconds, and the R0 of TB is quite high, so an untreated patient can infect 10 to 15 other patients. So the MTB life cycle is another one where it's really exploiting the long coevolutionary history it's had. So it uses a lot of our key effector mechanisms against us. So mechanisms that would control an array of other pathogens are actually utilized by TB to its advantage. So TB comes into your lungs, and now we're in the bottom of your lungs in an alveolus, and you have an alveolar macrophage that just hangs out in the alveolus and is there to eat pathogens and try to clear your lungs of the pathogen. So it's doing what it's supposed to do. It meets mycobacterium, and it eats it, and it takes it into an endosome and a phagolysosome, and it tries to kill it with those reactive oxygen and nitrogen species that we've been talking about and have come to know and love. And as it's doing that, it's gonna enter the blood vessels because it's potentially gonna try to go and report this infection to other cell types. But TB actually freezes its maturation at that point, and metabolically takes control of the cell, and the macrophage can no longer clear it. And so it's gonna still be able to report an infection to other cell types, and they're gonna come in and surround the infected macrophage. And this includes bringing in additional macrophages, these so-called foamy macrophages, and you actually get these syncytia where cells fuse together and become these large multinucleated cells, all surrounding this little locus of infection here. You have lymphocytes come in, CD4 and CD8 T cells, and they're all in this sort of stasis, sort of siege around the bacteria. It's very different than anything else we've seen. They can form a fibrotic cuff around it, and this entire structure is called a granuloma. And you can have this granuloma for the rest of your life with the cells inside of it actively alive and metabolic, but at a very, very slow rate. And the same thing with the TB, very slow turnover of metabolism. And it's this sort of stasis standoff, detente. And then somewhat stochastically, this is a really key question still of what reactivates TB. Certainly immune deficiency, so there's this active immune control here. It's not just everybody taking a time out, it's this active give and take. And so if you become immunosuppressed, for instance, if you develop HIV, then the bacteria will gain an advantage and break out of the granuloma, and you will cough, and you will spread the infection within yourself and to others. So as this spreads throughout your lung, you get more and more of these granulomas, and you get a fibrotic lung that is unable to breathe as efficiently anymore, and eventually will succumb to the infection in that way as the lung gets completely overtaken by bacteria and its associated immune response. So a lot of your innate immune system is able to detect different features of bacteria broadly and TB specifically. So some of the same TLRs that actually recognize malaria recognize some of the same lipid-rich compounds from TB. TB is such an old evolutionary partner of humans that probably a lot of these pathways evolved specifically just to detect TB compounds. And they're going to turn on that same inflammatory response that we keep talking about, TNF, IL-6, IL-12. It's early acute phase, fever, bring lots of cells to the site of inflammation, figure out what's wrong, and let's try to kill it because it's an intracellular pathogen. So we can drive this cytokine storm. So macrophages have responded. They've secreted a lot of these early innate cytokines. They'll bring in neutrophils, which will further amplify these chemokines and cytokines and will try to further eat at the macrophages. They'll bring in more uninfected macrophages and then lymphocytes, all with the goal of generating that Th1 response that gets rid of intracellular pathogens and will drive that macrophage to kill what's in its phagolysosome by making more reactive oxygen and reactive nitrogen species. So what the bacteria is trying to do is freeze that maturation of the phagolysosome before it gets fully acidified and dumped in with a lot of reactive oxygen and reactive nitrogen species, because those and the enzymes from the lysosome will actually kill the bacteria. So there are people that can kill the bacteria. There are infections that are aborted by your immune system. And it's really where the bacteria is able to gain metabolic control of this whole process and freeze it that determines whether or not the infection is successful from the bacteria's point of view or from the host point of view. And the bacteria can remain latent and grow. So the more activated a macrophage, the more likely it is to be able to destroy it. And so you can sense this evolutionary balance, because if we have super hair trigger macrophages that just kill everything and make tons of reactive oxygen species every time they see a piece of dust, that's not going to be good for us. We're going to develop a lot of inflammatory diseases. But if they're too sort of welcoming, then we're going to have a welcome home for TB. And you can see this, that TB actually actively prevents the maturation of the endosome. If you feed a macrophage just coated latex beads or live mycobacterium, the pH of that endosome drops to 5 within 10 minutes of the macrophage eating that bead, whereas it stops at about 6.5 when it eats mycobacterium. So it blocks the acidification of the lysosome. And so the persistence also requires other metabolic changes. So the bacteria turns on a completely different genetic program during its life in the macrophage than from when it's outside of the macrophage. Okay, so you've got this infected macrophage, which for the host at least conveniently is a professional antigen presenting cell. So it's got class 1, it's got class 2, it can present peptides efficiently. It's created a lot of cytokines, and it's brought in lots of lymphocytes, which are really tiny in this figure. And it's brought in non-classical T cells, gamma delta T cells, which are also really important for TB. They respond to some of those same compounds, like I said, that they responded to in malaria. And this is another thing that makes studying TB difficult in mice, because we don't have the same gamma delta T cells. And so CD4 and CD8 T cells, the regular T cells we've been talking about, also function to regulate the infected macrophage, either killing the infected macrophage or instructing the infected macrophage to try to kill or at least restrain the growth of the bacteria. And again, we're looking at these CD4 T cells. We're trying to get that type 1 immune response that's going to produce interferon gamma. And the thing that's going to work against the successful generation of those type 1 responses are the generation of these Treg responses, particularly these induced Treg responses. They're going to be trying to limit the damage that's occurring in the lung. I showed you these big granulomas forming where you could almost see the fibrosis. You actually could see the fibrosis in a lung of a person that had had TB for a long time. That's going to trigger this feedback regulatory response that's going to be characterized by the production of IL-10, and these opposing regulatory cytokines that are going to shut off the immune response. So the balance of regulatory and effector signals determine the activation milieu of the granuloma and the infected macrophage. And immune-associated pathology is also a risk, so we've got to have some regulatory balance. And IL-10 here is really the opposed cytokine to interferon gamma and really opposes lots of different cytokines. But we've got to keep a balance enough that we restrain the growth of the bacteria. The Th2 response would be almost entirely useless, although it does promote wound healing and tissue repair. But we're really trying to get this response and slight restraint from this response. And so this just summarizes a little bit about IL-10 and regulation of lung pathology. It's really the key regulatory cytokine. There's nothing else like it. It doesn't really have any sisters or brothers in the cytokine world. It's produced by multiple cell types and is required for survival in certain models of infection. IL-10 knockout mice often die very young because they develop these just crazy pro-inflammatory diseases from colonization by microbiome. And the pleiotropic effects of IL-10 are really not well understood at a mechanistic level. How IL-10 works to restrain immunity is not really understood. Even though we know the IL-10 receptor, we know some of the signaling it induces. How it shuts everything down is really at the molecular level not well defined. It's, I'd say, one of the really great mysteries in immunology currently. So this summarizes everything I've sort of told you so far, and really you can just see it right here. You recruit this group of lymphocytes that can be almost any type of lymphocyte to the infected macrophage, and you want all those lymphocytes to make interferon gamma. The macrophage will get that signal, and it will produce these reactive oxygen and reactive nitrogen species that will at least restrain, if not absolutely kill, the bacteria and prevent its reactivation and spread. Okay, so animal studies, like malaria, they're not perfect, but we can actually infect mice with TB. And this is sort of like that same graph I showed you for malaria, where we ask what happens when you knock out each of these pathways. So in wild type or gamma delta knockout mice, because we have the wrong gamma deltas in mice, you get this sort of set point of low levels of TB maintenance. Class 1 knockout, so that's going to eliminate CD8 T cell effects. You get this slightly higher level, so CD8 T cells do something, but not too much. Class 2 is going to prevent CD4 activation, so now we're really losing control of the bacteria. We get rid of the alpha beta TCR that gets rid of both CD4 and CD8 T cells. We lose even more control. It's basically this difference plus this difference. We get rid of NOS2, which is one of the key enzymes making those reactive nitrogen species. Now we're doing really poorly because we're not able to kill or restrain the bacteria inside the macrophage. So this is essentially affecting the macrophage's ability to restrain the bacteria. And just like in malaria, when we knock out interferon gamma, absolutely no control, no sort. It doesn't matter that all these cells are there trying to do other things. Without interferon gamma, it doesn't all come to anything. And so, HIV has shown the importance of CD4 T cell regulation in the control of TB. As you regain your CD4 T cell count in HIV patients that are put on antiretroviral therapy, TB incidence rates go way down, active TB, clinical TB. So restoring those CD4s and their ability to produce interferon gamma cause this improvement and recovery. And the HIV pandemic has caused 52,000, this was through 92, so obviously this has gone up quite a bit since then, excess cases of TB, and this was just in the United States. Okay, TB is one of the most fascinating and best studied diseases in terms of human genetics, and it really shows the role of interferons and why that mouse data is probably exactly correct in terms of predicting what's necessary for TB control. So the primary phenotype of individuals with genetic deficiencies in interferon gamma signaling is active susceptibility to mycobacterial disease. Sorry, the image here is covering up some of the words. And whereas deficiencies in type 1 interferons, so the interferon alpha and interferon beta, are more associated with viral susceptibilities. And so that's kind of shown here in this little Venn diagram. And the deficiencies in interferon gamma are associated with any mycobacterium, not just Mycobacterium tuberculosis. So it's a big family. And there's a lot of mycobacteriums that we don't worry about because they don't usually infect humans, because if you have any interferon gamma, you're able to control them. And so this is just a table of different immune deficiencies. So you can have profound immunodeficiency like SCID, where you're missing all your T, your B cells, your NK cells, and you actually can be infected with BCG, which is a live mycobacterium vaccine product. You can be infected with bird mycobacterium, Mycobacterium avium, Mycobacterium marinum, and of course MTB. But there are even more targeted deficiencies. So if you come down here, if you're just missing the interferon gamma receptor, you still show up with susceptibilities to all of these different very rare mycobacteria species. And that's generally how those individuals are identified, as they present with a weird skin infection that is diagnosed as one of these mycobacterial diseases that you would not normally see. And the immediate assumption has to be that they have a defect somewhere in the interferon gamma signaling pathway, either at the receptor, the cytokine itself, or the second messengers that deliver the cytokine. There are lots of attempts to make TB vaccines. One of the oldest vaccines that we have, which is BCG, which is this sort of live, not TB, but mycobacterial product, is still given and is thought to protect children. But there are more sophisticated recombinant products that are being worked on with I'd say so far limited success. So in the last couple slides here, I'm almost done. I know we're a couple minutes over. What really is the key correlate of your propensity to develop disease and severe disease with TB is how quickly you can mount that adaptive response and get a T cell to the infected macrophage to tell it to control that bacterial growth. And so if you can vaccinate animals or humans so that you have prime CD4 T cells that can get to the lung, or maybe even better, that are living in the lung, that can quickly get to the site of infection and start talking to that macrophage, it changes your bacterial burden set point. So this is at your set point if the T cells come in at day 20, if they come in at day 15, or if they come in even earlier. And that makes sense, I think. But again, this is something that is obviously very amenable to sort of a modeling exercise. So in the last 2 slides, I'm going to show you some data, and these are more from statistical models and machine learning type approaches to try to understand what controls reactivation. And this is still a sort of a burning question. This is a very prominent paper from about 10 years ago, but what it found is still something that is a topic of a lot of study. And so there are large cohorts of TB-infected individuals that are followed to try to understand how to prevent reactivation, when treatment needs to be started, and what the most effective means of treatment would be. And so it's difficult to follow these because the diagnosis and assessment of TB still relies on chest radiography. And so what this group did is they started sequencing PBMCs, because the idea was that we could actually look at PBMCs instead of having to do these chest radiographs, which are obviously significantly more difficult to do. Maybe we could find something that's a predictor of whether or not somebody was going to reactivate TB. And then these also can generate hypotheses for a better mechanistic understanding and maybe modeling what actually happens immunologically in an otherwise healthy individual that then mounts TB reactivation. And so this is what they had, and this is very typical now, you'll see, of a lot of human studies where you're trying to find a biomarker or classifier. And initially the goal in these studies is usually to find just a marker, a correlate, because that would be clinically useful. But then you also, from the immunologist side, hope that that correlate tells you something mechanistic, or at least gives you a hypothesis to test mechanistically. So the setup is usually that you always need, particularly if you want to get into a journal like Nature and really have an impact on the field, you want a test set and a validation set. So you want 2 different sets where you first develop your classifier and then you retest it without using any of the same individuals. So you've got a group of people all with latent TB, and then you also maybe have a positive control here of people that have reactivated TB. And you what they've done is just a very simple type of classification where it's basically just clustering based on gene expression, unsupervised clustering. And what they found was that there was a group of latent individuals that clustered with the active cases. This little group here that clustered with the active cases. And they made a classifier out of that, and they applied it to another group in South Africa, and they found the same thing, that this latent group clustered with the active group. And so they were able to detect that these latent individuals that would then go on to activate TB could be detected a few months before they could detect them with the standard clinical tests. And so that might be very useful in terms of quarantining individuals and treating them aggressively to potentially limit the effects of their reactivation. And what it turned out that gene signature, what that gene signature actually correlated to was neutrophil activation. So it was just a very clean signature of an enrichment of neutrophils in the blood prior to the activation of TB. And this is where human studies and immunology gets very hand-wavy, because you can imagine a dozen different hypotheses to explain why neutrophils were appearing in the blood in individuals about to reactivate TB. It could be that maybe they had some other bacterial infection, and that was then distracting, which was recruiting neutrophils and then distracting the immune response from dealing with the TB. That's a plausible explanation, testable, to look at that. But it could be that actually the TB had already started to reactivate, and the system was mobilizing neutrophils to go to the lung to try to deal with it, but it was an activation level below our clinical level of detection. And so maybe those neutrophils were going to see TB, maybe they were going to see something else, maybe a third hypothesis that we could easily come up with. But this type of data is now extremely useful, and I think it's sort of a recurring pattern you see in immunology, where we extract from these high-dimensional data sets a relatively simple and straightforward correlate that we then try to turn into a mechanistic hypothesis for a biological outcome. Okay, so the summary here is that in most individuals, MTV is never completely cleared following initial infection. The primary effector mechanisms are macrophage bactericidal functions, but their success is determined by the cytokine, particularly interferon gamma, and cellular regulatory environment. And small, subtle shifts over time or dramatic short-term changes can lead to reactivation and disease. So I just saw the question that popped up. If there was a successful vaccine for TB, would it be useful as a treatment as well as preventative because of the latent nature of TB? Exactly. I think that a lot of the vaccine trials that have already started have been done in these latently infected individuals because they're adults. Generally, we like to do vaccine trials in adults first rather than kids in order to get safety signals. But if you could start therapeutically treating adults, that's also a way of trying to interrupt spread and reduce the disease burden worldwide.